I am Edward Dennis, and I welcome you to the second part of Lecture 5, known as Lecture 5B on Specialized Lipids and Disease. In Part A, we discuss sphingolipids. In Part B, we will now focus on lipids in energy metabolism, and first introduce and discuss ketone bodies, which relate strongly to diabetes. We will then integrate the various aspects of lipid metabolism that have been covered in this course. We will start with an overview of ketone bodies and then look at the discrete parts individually. Basically, the breakdown of sugars, fats, and amino acids, that is glycolysis, fatty acid oxidation, and the degradation of some amino acids, lead to acetyl-CoAs. When they accumulate, they are converted into HMG-CoA by a synthase and further converted to free acetoacetate. The acetoacetate can be interconverted to beta-hydroxybutyrate enzymatically and non-enzymatically to acetone. Because of the presence of a ketone group in acetoacetate, these three compounds have been referred to as ketone bodies. The ketone bodies are synthesized mainly in the liver, but then they can be transported to peripheral tissues where, if they're not excreted by the kidneys, can again be interconverted to one another, and the acetoacetate can be broken down further to acetyl-CoAs, where they may result in fatty acid synthesis or in oxidation through the TCA cycle. Acetate is made mainly during starvation conditions and involves three enzymes. Two acetyl-CoAs are condensed by an enzyme called thiolase to make acetoacetyl-CoA, and then a third acetyl-CoA is added by the enzyme HMG-CoA reductase, which stands for hydroxymethylglutarol-CoA synthase. The HMG-CoA is then broken down by HMG-CoA lyase to produce a free molecule of acetyl-CoA and acetoacetate. The acetoacetate can be converted to D3-hydroxybutyrate by a dehydrogenase. This enzyme is basically at equilibrium and can serve to interconvert the levels of hydroxybutyrate and acetoacetate. Non-enzymatically, a small amount of acetoacetate is decarboxylated, leading to acetone. Acetone itself is very volatile, and the excess of the acetone, which is very insoluble, is excreted through the lungs and can often be smelled on the breath of an individual who has built up ketone bodies, most particularly acetoacetate. Ketone bodies are synthesized in the liver and are then transferred to peripheral tissues which have an energy need. The acetoacetate can be converted to acetoacetyl-CoA in these peripheral tissues by an enzyme called 3-keto-acyl-CoA transferase. This acetoacetyl-CoA can be further broken down by thiolase to make free acetyl-CoAs. And these acetyl-CoAs can enter the TCA cycle in mitochondria to be oxidized and generate ATPs, or when there's an excess, they can enter the fatty acid synthase system in the cytosol of cells. The synthesis of ketone bodies in the liver is not exactly the reverse of its breakdown, which have a different set of enzymes and function to utilize the ketone bodies made in the liver as needed. Now we wish to put the ketone bodies in perspective of lipid metabolism. Complex lipids, carbohydrates, and proteins are broken down to their simpler components, fatty acids, glucose, and amino acids. And these in turn are broken down by catabolic processes to acetyl-CoA. The acetyl-CoA is generally used in anabolic processes, converted to malonyl-CoA, a key component in making fatty acids. And the fatty acids that are made are converted and stored as complex lipids in the form of neutral lipids like triglycerides, phospholipids, sphingolipids, and as the esters on cholesterol. When energy is needed, acetyl-CoA is fed into the TCA cycle through citrate, where it results in the production of CO2 and ATP, which is used for energy. In certain physiological conditions, principally starvation, that acetyl-CoA or acetyl-CoA in particular is converted into HMG-CoA and forms ketone bodies, which can be transported and further broken down to acetyl-CoA. HMG-CoA is also a precursor for the synthesis of sterols, including cholesterol. It's important to keep in mind that the oxidation of fatty acids, which results in acetyl-CoA units, occurs in the mitochondria of cells, whereas the synthesis of fatty acids, which utilizes acetyl-CoA, occurs in the cytosol of cells. Once the simple fatty acids like palmitic acid are made, the further elongation and unsaturation of these fatty acids occurs in the endoplasmic reticulum at various subcellular membrane locations. And these fatty acids that are elongated and unsaturated are incorporated into neutral lipids like triglycerides, phospholipids, and sterols. It's Important to recognize the specific intracellular localization and compartmentalization of the biological processes we've been discussing. It's equally important to recognize the specificity of the organ localization. The liver is at the center of metabolism, and it is very involved in the breakdown of metabolites and their oxidation. It sends out glucose to the brain or brain function, and also under conditions of starvation, ketone bodies. The liver also makes lipoproteins, which contain a large amount of triglycerides that are produced and delivered to the adipose tissue. And when energy is needed, the adipose tissue is activated to release free fatty acids that return to the liver. The glucose and fatty acids, and under starving conditions, ketone bodies produced by the liver are also sent to muscle, heart, where they are the essential components that drive their metabolism. And the molecules that are no longer needed or are oxidized are sent through the bile duct to the intestine for excretion and some recycling. The blood is an important organ because it transports and contains the triglycerides as part of lipoproteins. The fatty acids are generally bound to albumin, where they travel around the bloodstream. Glucose is freely dissolved and picked up by peripheral tissues, as are ketone bodies. Blood is one of the few organs that are accessible for analysis in humans. And if one compares the human plasma metabolites, we see that about 3/4 of them are lipids, and the other biological molecules, nucleic acids, amino acids, and sugars, make up the remaining quarter. The lipids, about half of them are sterile lipids, and the other 5 categories of mammalian lipids constitute the other half: sphingolipids, glycerophospholipids, prenols, glycerolipids, and free fatty acyls. There is an enormous diversity of lipid molecular species in human plasma. Shown here is the notion that acetyl-CoA is converted to fatty acids for storage, and the free fatty acids are esterified in complex lipids like glycerolipids, glycerophospholipids, and sphingolipids. Some of the fatty acids are converted to oxidized forms of polyunsaturated fatty acids known as eicosanoids. In addition, fatty acids are esterified to sterile backbones, principally cholesterol. Now, acetyl-CoA, in addition to being a precursor of fatty acids, can be converted to isopentyl pyrophosphate, which is converted further into the prenyl backbones of the prenols and of the sterols. So all the lipids are really derived from acetyl-CoA by biosynthetic processes. Shown in orange circles are the number of distinct lipid molecular species for each of these categories of lipids that were found in abundance in standard human plasma samples. In summary, lipids are very central to many diseases, and in particular, the metabolic syndrome. And these numerous lipid molecular species discussed on the previous slide are involved in the sequelae of these diseases. Many of the lipids, as pictured here, are parts of lipoproteins, which play a critical role in the accumulation of lipids and their transport. Shown here is the role of lipids in obesity, in adipose tissue, their importance in insulin resistance and diabetes, and the effect on various cardiovascular diseases, including atherosclerosis, thrombosis, myocardial infarction, and stroke, and as a general component in inflammation. Understanding the metabolic syndrome and metabolic diseases requires a thorough understanding of the lipid components and their role in metabolism and disease.